get it. Okay. So I am recording the session from here on. Okay. So we were supposed to understand and learn the zinc finger technology. The second stair is the Talens, and the third stair is the CRISPR-Cas9 system, which is now being used as a tool for targeted genome editing. But even before that, last night I was thinking about your lecture, and I thought that even before the zinc finger, let me tell you another background of that stuff, from where actually everything started. What were the old-fashioned methods to edit the genome, and how it was done in earlier times. Okay, so all set. Just let me explain you a little background before actually entering into the three techniques which I wanted to teach you. Whenever we want to, I must say, change the genome. Think about the DNA here. This is the DNA which is present within the nucleus. Okay, which is present within the nucleus. The blue color DNA, human genome, it's a part of any chromosome, and we want to edit this one particular portion on our genome. And I have already explained this thing that targeting one particular base pair in the genome is quite enormous to imagine. It's very small as compared to the total size of the genome. So we want to make changes here. We want to introduce a gene at the place of our own choice. So what were the tools before the zinc fingers and the Talens were there, where people were using in order to integrate an external gene at the place of our own choice? You know, you have studied in the gene therapy that we can send the thing and the external gene, and that external gene might incorporate anywhere within the genome. Multiple copies might be incorporated, but we never know that where that gene will actually end it into the genome. We only came to know about the phenotype after that. If we start receiving the new functions, we can successfully claim that we're able to send the gene within the genome. But we were always failed to understand that where actually it landed within our genome. That was the question, and people have tried to fix that problem with the help of a very wonderful phenomena called homologous recombination. That phenomena is called as homologous recombination, or more commonly called as HR. The process of homologous combination is that it involves a vector. Please stay with me. Okay, it involves the vector. That vector contains a certain fragment, a certain cassette of the DNA, which is used to incorporate or to send the gene of our own choice at the place of our own choice. This is like adding more complexity to the thought. Just stay with me. If you find any trouble, just let me know. Okay, feel free to speak. Do not feel shame if you do not understand it correctly. Okay, this is very critical, very important. That will going to help you guys to feel better. Okay, as a university student, just stop me if you feel I don't mind spending more time in these things. So there is one portion of the vector which is utilized to send the gene of our interest. The vector was designed in such a way that along the sides of the gene of our interest, or the gene which we want to send, we created two homologous fragments of DNA. This is the one homologous fragment of the DNA. This is another homologous fragment of the DNA. And we place the gene of our choice, which we want to send, inside these two homologous arms. So this is the gene, a new gene we want to send into a host's genome. And we also want that this gene should land and should incorporate at the place of our own choice. And what is the place of our own choice? This is the place of our own choice. That is the place of our own choice. So this is like the agenda of our initial conversation. These two arms are created in such a way that the sequence of DNA here is 100% similar to the sequence of the DNA present here. Okay. So I can refer it as a left arm, okay, as L. So this sequence is 100% homologous to that part. And this portion of this and the sequence of DNA here is 100% match with that portion of the DNA. This is called as a as a right arm or the right homologous fragment of the vector. So we design a vector in such a way that two big regions of the DNA matches with the host DNA. The arm length is usually in hundreds, okay, not in base pairs, it's hundreds, like 500 base pair, sometimes 700 base pair, sometimes 1000 base pair. You can see Google and internet that people have created, like try to be more longer. The longer the similarity, the more chance of integration is there. And it includes an external portion, that one, the gene which we want to send here. Now the first problem or the first task is that we should send this vector, okay, this vector. People are coming, still coming. The first task is to send this vector right within the nucleus. Right arm and the left arm. So this is like L arm or this is the R. And you know that there is a region between this. This is a gene we want to send. That's an external gene. The first job is to send that stuff right in front of the human genome, right in front of the host genome. This is the first task. And that is where most of the technology were failing at that time. Because the vector we are sending into the cells, it will not going to infect all the cells. It will not going to enter in all of the cells. Even if they managed to enter into the cells, it was destroyed in the cytoplasm. Very few of them able to penetrate into the nucleus. And that is why the efficiency of sending the vector into the nucleus is very low. And that was the problem with the gene therapy at that time. So we have a problem of efficiency. That is why we ended up with like 1 to 2 to 3% of the cells which has host genome together with the vector. It's a very big problem actually for the researcher and scientists. So very few of them able to send that stuff here. That's the first thing. Now what will happen after that? How that stuff is going to incorporate. Remember, this part is 100% match to this region. This part is 100% match to that region. So how it is possible that this stuff is going to be incorporated here? If you remember, the sign and the lines and the LTR stuff in the lentiviruses, they were using, you know, transposons and jumping genes and similar sort of activities of the human DNA elements to get themselves incorporated into the host genome. That was quite untargeted. Okay, that was not targeted stuff. You never know. But this is the targeting situation. You are not using the lines and the signs and the LTR things or jumping genes or transposons to send that external DNA because you want that thing to be incorporated at the place of your choice. So this is a very big question. Homologous recombination was the only thing people were using at that time to send the things at the place of their choice. But you know, already the efficiency was very low. And now the probability is much less. What will happen? Can anybody guess how it is possible that this thing will going to incorporate here? Shabash. Please speak or raise your hand. I want what your brain is thinking. If you are with me, I know it is very difficult to stay alert at this time. Ji Aisha.
So are we using restriction enzyme?
We are, Aisha is saying we might be using restriction enzyme. Answer is no. We don't want to cut the genome of the host cells. Okay, we are not sending the restriction enzymes. If we send the restriction, You know, the restriction enzyme concept which you are carrying is related to the experiment in which you have an extracted DNA. If you have an extracted pure DNA and then you give the restriction enzyme, you can cut the fragment of DNA at the place of restriction enzyme sites. But here, imagine this is a cell. You are sending the vector. If you send the restriction enzyme over there, that will going to, I mean, it is not possible to send proteins, by the way, from outside source right within the nucleus. Abhi. But I'm happy that you speak about it. Okay. So, Abhi.
Sir, maybe during cell division, homologous recombination takes place normally, so maybe this becomes a part of it.
That is the question, ना कि ये homologous recombination अभी हम इसी की logic समझने की कोशिश कर रहे हैं ना. I want to test your knowledge, not knowledge but your thoughts. It is okay. You can आप कोई भी बोंगी मार सकते हैं. I don't mind. But just speak. Anybody else?
Sir, maybe the complementary regions in the left and right arm will bind to the DNA sequences in the host DNA because they are 100% similar. And once the binding of the left and right arm occurs, the fragment in the middle, that is the gene of interest, will also integrate into the genome.
Yes, this is homologous recombination. And my question is still there. I mean, this is, remember, this is the double-stranded DNA of you are sending. This is the host double-stranded DNA waiting there. And I mean, ये चिपक जाएगा इसके साथ जाके. क्या करेगा इसको, क्या करेगा, काट भी नहीं सकता, निकाल क्या, I mean, आपके दिमाग में क्या आ रहा है यहाँ पे? शाबाश.
Sir, during replication.
ये किसने बोला? ये किसने बोला है? राबिया, शाबाश. राबिया दूर की कोड़ी ले. Wonderful answer. Shabash. That thing will going to wait there for the process of replication or the DNA repair mechanism. The DNA repair mechanism, when there is a destruction in the DNA, it tries to fix the things. But that will not going to occur. That will not going to happen. We are not destroying DNA. The DNA is there. So the system will stay. The vector should survive to the time when the chromosome will start the procedure of DNA replication. That is the time when the single-stranded become, the double-stranded will become single-stranded at the time and the DNA will incorporate it. That was the time when these things, the DNA replication machinery will start using that vector fragment and will start incorporate that vector into the host gene. That's the only chance. But think about the probability. The sending the vector is the trouble. Sending the vector to the nucleus is the trouble. Allowing that vector to survive at the time of replication is the trouble. And then replication machinery should be able to find these two things together. Because replication machinery includes a lot of things. That includes, aapne suna hoga, restriction enzyme that can cut the DNA. That includes exonucleases, which can destroy the extra and unwanted DNA fragments. That includes the DNA ligases, which connects the DNA. It contains all the package. So these things will going to be incorporated if the process of replication is there. So if you put all these things in perspective, and if I ask you that using the homologous recombination is a way, is there any chance for the external DNA to become incorporated? You can guess the product. The product will be like this. This is the human genome, and this will going to be the L side is replaced. The L size of the human genome is replaced. The R side of the human genome is replaced, and together it will going to incorporate the gene of your choice. And of course, the people who design these cassettes, they not only send the genes, they also design the promoter region as well, so it can start working at the same time. The efficiency of the process will be like, it's like mind-boggling. Nothing. It's like you are doing thousands of experiments and then you might be able to get this wonderful combination, all of these factors together in one experiment, and you might be able to have your gene at the place of your own choice. So what will be the big question mark on this procedure? And that question mark is about the efficiency. But people do find a reason. If there is a place where you need to go, you can go there on aeroplane. Aeroplane is not there, you can go there by train. The train is not there, you can go by car and then by other means, and then on your foot. If I ask you, can you go to Karachi on your foot? The answer is yes. But the efficiency and the probability is like almost, impossible. The same goes for this process. So the people were wondering, they are, banging their heads because of the low efficiency of homologous recombination. They thought that they might be able to answer the critics who were like troubling in terms of gene therapy, the problems in gene therapy. They might address this thing with the help of homologous recombination, but that was not addressing the things. So what they have done, they came up with the new things using the DNA binding proteins. Remember, zinc fingers, they are the DNA binding proteins. They engineer the DNA binding proteins. DNA binding, what are the DNA binding proteins? Our DNA has bounded with lots of different types of proteins. Transcription factors. There are DNA binding proteins which are controlling the three-dimensional architecture, topology of the genome, controlling genes, expressions of many genes. They are creating active and inactive domains. So a strand of DNA is nothing, is nowhere near to life. The thing which actually give life to the DNA are the DNA binding proteins present on the DNA. Now, people were working on these things and they came to know that a DNA binding protein knows where it is supposed to bind on the DNA. So they do have some, similarity, and very famous DNA binding proteins you guys have already studied. Restriction enzymes. Restriction enzymes like have a DNA binding domain, and then they have like the scissor. If I can draw the scissor perfectly, I don't know they look like scissor or not. So one side of the restriction enzyme, they act like a nucleases. They can cut the DNA. And the other side of the restriction enzyme side has a domain which can recognize a particular sequence of the DNA. So this is called the DNA binding domain. This is the DNA binding domain of every restriction enzyme. And then there are scissors over there. So that is why restriction enzymes are very specific. They can recognize one particular sequence of DNA. Scientists were studying about the DNA binding proteins, and they came to know that lots of different types of DNA binding proteins other than restriction enzymes were there. And one of the most famous of all times were the zinc finger proteins. The zinc finger proteins have the speciality that they can recognize three nucleotide sequences. Please remember these important words. Okay, one zinc finger protein. They are called as zinc finger proteins because you can say their three-dimensional structure is stabilized with the help of a zinc ion. Zinc ion is there in their structure. That is why they are called as zinc finger proteins. And the zinc finger proteins, they are able to bind or recognize three nucleotide base pairs. Typically, these three nucleotide base pairs should start with the G. So this is like a limitation for the zinc fingers as well. So people were studying about the zinc finger protein, and they came to know that there are lots of different types of zinc finger proteins. They can recognize three base pairs in sequence, and these three base pairs should start from the G. They recognize DNA sequences in triplets. So hundreds of zinc finger proteins are out there. How about connecting different zinc finger proteins which are recognizing different sequences to create a zinc finger array, of our own choice? For example, this one zinc finger protein is able to recognize GGG. This one can recognize GGT. This one can recognize GAC. If you engineer them together, if you connect these three zinc fingers together, they will going to create a zinc finger array which can recognize 9 base pair sequence on the DNA. If you check the probability of having 3 base pairs on human genome, you will end up in millions. If you try to check the probability of having a 9 base pair in the human genome, you will find few. And if you're able to add one more zinc finger over there, having a 12 base pair DNA sequence again in the genome is almost none. So that increases the specificity of the protein. So now we have an engineered protein made up of 3 zinc fingers, which is able to recognize 9 base pair target on the gene. If we design and select the zinc fingers smartly, we can target any region on our genome. So that's we have achieved. We can create a zinc finger array which can go and bind itself with the place of our choice. All you need to do is to find out the right and appropriate zinc finger. So now we have created the zinc finger domain of our own choice, which can bind itself with the DNA sequence of our own choice. You will come to know why we are doing it, because please remember my initial talk. That will going to help you. So after that, we have incorporated a nuclease. We have incorporated a nuclease. Nuclease is the one which can act as a scissor with this zinc finger array. Usually the nuclease which is used for this purpose is called as FOC1. You can ask me why FOC1 was used, why not other, but they have used FOC1 for that purpose. So that is how they able to target one particular place on the genome. So that stuff can find the place of your choice, and this thing is a nucleus that will going to destroy or cut the DNA at the place of your own choice. Please keep this question, why we need to cut the DNA, okay? And people have engineered the zinc finger array like 1 zinc finger array, 2, 3, 4, 5, and you can answer in your GRE subject test or in an interview that a 4 zinc finger module will going to target almost 12 base pair on the genome, and that is sufficient. So people have engineered that stuff. This is the paper. You should go and see how they have created. And they incorporated the FOC1. That is a very important question. Why the FOC1 nuclease? Why the FOC1 nuclease? Because the FOC1 nuclease work in dimers. Remember, it works in dimers. So if you engineer a zinc finger array, bind it with a FOC1, that will go and bind with the host genome, but that will not going to create and cut the DNA. That will not going to create a double-stranded DNA break until unless it find its partner, the other FOC1. How you can send the other FOC1? That's another question. You need to know your target location of the DNA. So for example, this is the this is the target DNA, and you want to send and create a double-stranded DNA break at the place of your own choice. You need to design 2 zinc finger modules. One which should be able to bind here on one strand of the DNA, the other which should be able to bind here on the other strand of the DNA. This one includes the FOC1. This one includes the FOC1. You need to design this module in such an efficient manner that they should be able to present at a specific distance from each other. So these 2 FOC1s should face each other and should be able to create a dimer. Once the FOC1 is dimerized, it will going to induce this double-stranded DNA break here. So 1 zinc finger array should be sent to the cells. And you know how the zinc finger array is sent? Zinc finger is created in a vector. A vector sequence is created. That vector is sent to the cells. it expresses the messenger RNA. That messenger RNA will going to create this protein. They should also send another vector. That vector will create another messenger RNA, and that messenger RNA will encodes this zinc finger module. So 2 vectors should be sent to the cells. The cell will utilize those 2 vectors, create 2 zinc finger modules, designed smartly in such a way that it will bind to the opposite strand on the opposite side of the place of your choice, allows the fork one to dimerize, and that will cleave the double-stranded DNA. It will create a double-stranded DNA break. And that was the target of the scientists and researchers at that time. You can ask me why. So the complexity in this procedure, you can guess what? You are required to express 2 vectors at the same time in one cell. So sending 1 vector and pushing that 1 vector to express is a problem. Sending 2 vectors at the same time, pushing these 2 vectors to express at the same time is even worse. That will actually, every step will going to decrease the efficiency of the procedure. So you are putting and sending these 2 vectors together within the cells, asking the cells to utilize these 2 vectors, producing 2 zinc finger modules, and these 2 zinc finger modules will then going to bind at the place of your choice. This is another level of inefficiency, that if they fail to bind there, but they will going to produce a double-stranded DNA break at the place of your choice. If everything works well, these zinc fingers will be able to produce a double-stranded DNA break. The specificity is very high because you are actually targeting a very specific location on the genome, and you're able to produce a double-stranded DNA break as well. Neha.
Sir, is the gene of interest present in the same vector that contains the messenger?
No. अभी हमने कोई gene of interest बताया नहीं. अभी हम सिर्फ क्या कर रहे हैं? Producing a double-stranded DNA break. And anybody else? Okay. But you are thinking on the right side. This is another question. What we are studying, targeted genome editing. So where is the? So at least we know how to create a double-stranded DNA break at a place of our own choice with more efficiency. That we can do. This technology can be used to create a knockout. Am I right? Knockout, because you can destroy the gene. You can send the zinc finger modules in such a way it can go and clip the p53 gene. It can destroy the gene of your, because once the DNA double-stranded break is there, what will happen after that? You know what will happen? The DNA will try to repair it itself. DNA will try to repair it. Double-stranded DNA break is a havoc, is a hurricane within the genome. Genomic instability aa jayegi. Fatafat wo kya karega? It try to pick the things and repair the DNA. You can see my another lecture in molecular biology about DNA repair mechanisms. It will try to pick these 2 things and try to connect it. For that purpose, they will incorporate some base pairs. Sometimes they cleave some base pairs, and they will going to put them together and try to fix it. But in that process, the DNA integrity will be lost. In that process, because it added few base pairs, it might remove some fragment in order to fix it. Yes, it is jagah pe diwar toot jaye. The mason will try to remove lot of portion to fix it perfectly. Similarly, that portion will be fixed. The natural DNA repair mechanism will be activated, and that will going to fix that thing. But during that fixing process, some portion of the DNA might be lost. And that will lead to the dysfunction, that will lead to the trouble and mutation and loss of function of that portion. And you can introduce a gene knockout at the place of your own choice. So jitne bhi gene knockout hue hain, initially report hue hain, p53 knockout mouse, flana wala knockout, all the knockout mouse which were originally created, that were used by this technology. It actually helps us to understand the function of many genes. Now what happens after that? Once the DNA double-stranded break is there, because of the zinc finger nucleases, there will be 2 mechanisms which will be adopted by the cell, by the DNA repair mechanism. The first is the error-prone repair. In the error-prone repair, I told you that the cell will try to connect these two things together, and during that process, it might clip some fragment of the DNA. Sometimes it incorporates few base pairs to connect these two things together, and that introduces a mutation, that is the genome. So if you are interested in creating a targeted mutation at the place of your choice, this technology is good. But if you want to add an external gene, which was your original interest, then you need to choose another natural phenomena. Then you need to harvest another natural phenomena. And that's what we call as homologous recombination. So you have to do two big things at the same time. First, you need to send the zinc finger nucleases to create a double-stranded DNA break at the place of your choice. See the red color, left side and the R side, at the place of your own choice. Then you need to create this donor plasmid. You need to create this donor plasmid. The donor plasmid has the L side as well as the R side, similarly as I tried to draw in the earlier part of the lecture. And it also contains an insert, the look at yellow one, the gene of your choice, which can incorporate. Think about if the zinc finger nucleases are not there. Think about in the absence of this thing, is it possible that this donor plasmid is going to be incorporated? Yes or no? In the absence of the left side of the slide, is it possible this donor plasmid will be incorporated within the genome? In even slightest of the possibility is there?
Yes, sir.
Thank you. The answer is yes. But as I explained in my earlier slides, the process will be highly inefficient. Highly inefficient. But what if we introduce a double-stranded DNA break? Because if we introduce a double-stranded DNA break at the place of our own choice, the DNA repair mechanism will start working immediately and will start trying to fix that thing up. And if a donor plasmid is there, like this one, if a donor plasmid is there, the DNA repair mechanism will going to see a similar sequences. That is the first priority. If you ask me, what is the best way to fix an error is to put some already available construct at the place of the trouble. The DNA repair mechanism will try to fix the double-stranded DNA break, and if a donor plasmid is there with the similar sequences, L side as well as the R side, excellent information, excellent bait for the DNA base pair machine, DNA repair mechanism. The DNA repair mechanism will come and cleave this thing out and use this similar sequence and incorporate that fragment at the place of your choice where you have incorporated the double-stranded DNA break. So the process of homologous recombination, if the efficiency was like nothing, now the efficiency is something like 3 to 4 to 5% efficiency now. But now think about the practicality of this exercise, how practical and difficult that thing is. To execute a DNA double-stranded break and the incorporation of the gene of your choice. You need to send a vector, not one vector, actually multiple vectors, to deliver the DNA, to deliver the machinery which can produce zinc finger array into the cells. First thing, that will going to introduce a double-stranded DNA break. At the same time, you should give a donor plasmid which has a similar arms, and DNA repair mechanism will then do the rest of the job. it will going to utilize this donor plasmid and incorporate it into the DNA. And how laborious that will be. कैसा feel हो रहा है सुन के? Laborious है नहीं? So your instructor have done this thing during his masters. उस वक्त ये बड़ी मशहूर टेक्निक हुआ करती. So that stuff, the zinc finger assembly has actually reduced the time and efficiency and increases the efficiency a lot in respect to the homologous recombination method. So that is why people were started thinking that I mean that is the future. I mean we actually enhance the capacity. Now practically we can introduce at least the gene knockout comfortably. And look at this data and you might be able to understand this thing, that this is the sequence of the wild type sequence of the genome of the mouse. And they designed the zinc finger array, and you can guess one of the zinc finger array binds somewhere here, and then there is a fork one there. The other binds to the other strand here, and then the fork one there. Then these two fork one dimerize and clip the double-stranded DNA here. The DNA repair mechanism then tried to fix this thing and ended up in this sequence in one cell, this sequence in another cell, this sequence in another cell, this sequence. All these are mutant after treating with the zinc finger nucleases. So that means the scientists were successfully able to induce mutations or destroy the function of genes at the place of their choice. That is how they have created the P53 knockout rat at that time. It's a 50% possibility of that rat, it will going to develop the cancer during its lifetime. And that was like an excellent model to study science, to study cancers after that. So after that, you can analyze the host genome. They have that for the very first time they tried to analyze the host genome at the place of their own choice, and they found the mutations. So it was very, I can tell you, fascinating at that time that they able to introduce mutations at the place of their own choice. And you should be able to understand the reason that why they use fork one, because binding of one zinc finger at one side is not good enough. You need two zinc fingers to bind at the place of your choice. Only then it will be able to create a double-stranded DNA. That increases the specificity. And even then it is possible that you might find similar sequences and zinc finger nucleases might bind somewhere else and might introduce mutations at the place you don't want to. And this is what we call as off-site activity. This is what we call as off-site activity. And that will create a chaos in the cells. And that is not nice. So that zinc finger nucleases increases the efficiency, but does not manage to stop the off-site activity as well. So the off-site activity is still there. Ji Neha, aap question karen?
Sir, we know that there is a possibility that the 3 plasmids we are sending might go to different cells and not one single cell.
Yes, yes.
But during vector construction, do scientists incorporate any sequence that ensures that they go to the same cell, or is it completely random or based on chance?
They tried to create reporter things. They tried to use GFP to tag one of the vector, RFP to the other, so that they came to know that whether these two are actually incorporated or not. But that was quite, I can tell you, difficult. I mean, it actually reduces the efficiency. The more variables you will have, the more chances that you will going to lose the efficiency. But they have tried to add tag them with certain proteins which they can check whether the zinc fingers are entered successfully. They tried this thing in steps as well, but all you have to have all these things together at one place. But nowadays this is not a technology anymore, but this information is absolutely necessary to understand this branch of science. It will going to develop your mind It's a very big lecture, I can tell you. Targeted genome editing comprises many classes, and this actually absolutely necessary for you to build the correct concepts for this part of science. You should understand the troubles क्या थी, शुरू कहाँ से हुई कहानी, efficiency factors जो है वो, because all these things are later on addressed by the upcoming techniques. Now अभी मैंने आपको Talens और CRISPR नहीं पढ़ाया है, but you should be able to understand that the why CRISPR is better than the zinc fingers. Maybe it has addressed the efficiency factors. Maybe it addressed these multiple vector problems, efficiency issue, off-site activity troubles, and more site specificity. All these things you need to acquire, you need to develop, you need to be very specific, no off-site activity, increasing the efficiency. If you have all these three things together, you have a wonderful system. And that is why maybe after that, people feel comfortable of having, manipulation in the genomes. Because until you don't know about everything, you're not sure where you are actually editing, you are not in control of the genome. Like if a textbook is given to you and somebody asks you to amend that textbook to make it better, you should be able to incorporate things at the right places. If you put a sentence in a wrong place, that will not going to improve the textbook, that will going to hurt the meaning of a certain paragraph. That paragraph might be very important. So the more you have skills on these techniques, the better it will be for you.